Okay, hello students. In this video, we're going to go ahead and start the 5.2 notes where we're going to continue talking about genetics and inheritance, but on a slightly more complicated level. In this video, we're going to look at topic 1, where we're going to go over some non-Mendelian inheritance. So non-Mendelian inheritance refers to situations that where genetics is playing out in a way that Mendel did not observe or figure out was possible, albeit Mendel was just working with pea plants, and so he was very limited in what he was going to figure out, but he did discover definitely the basics of how genetics works. But we have found since then, with the discovery of DNA and chromosomes and meiosis and lots of other examples in different organisms of many situations where things are going to play out a little differently than what Mendel would have thought. And so in these notes, there's basically five things in this video, in this topic, that we're going to talk about that are kind of a little bit different than what Mendel originally came up with. The first thing is the fact that there's different degrees of dominance that occur sometimes for specific genes. And so for Mendel, he came up with the fact that, or he discovered that there was dominant and recessive alleles. And so sometimes when you ended up with two different alleles, phenotypically you would just show evidence of having just one of those alleles, like one of them would win, it would be the complete winner. So if you had, for flower color, if you had a purple allele and a white allele, if purple was dominant, you would have purple flowers, and they would be just as purple as any other purple flowered plant. That's what we call complete dominance. And for most of, or all of the traits that Mendel was looking at, there was a completely dominant and recessive allele, where one was always completely winning. But what we know since then is that there's many situations where there's alleles that are different, that are not, where one of them is not completely dominant over the other. Two examples of that are incomplete dominance and codominance. In incomplete dominance, that is when one of the alleles, neither allele is dominant over the other. And if you end up with both of them, two alleles that are incompletely dominant, then you're going to end up with a phenotype that's some sort of blended version of both of those alleles. So for example, in flowers, let's say there's two alleles for flower color, red and white. If you end up heterozygous, if you end up with a red allele and a white allele, and they're incompletely dominant, let's say the red allele and the white allele are incompletely dominant, that means that you're not going to be red and you're not going to be white. They're both going to lose. Okay, there's going to be no winner. Instead, they're going to be some compromised blended phenotype that you're going to show instead. In this case, it would be pink. So if you're heterozygous, you would have pink flowers. Not red, not white, but some sort of compromised middle ground between those two alleles. So that's what incomplete dominance is. Now, codominance is different, and that is when you, for some genes, there are situations where two different alleles are sometimes both dominant, so we call it codominance. So for, again, let's use the flower color example. Let's say for a certain type of flower, there's red alleles and white alleles for flower color, but this time, let's say they're codominant. If those two alleles were codominant, that means they're both actually going to win in terms of what phenotype you're going to have. If you had red and a white allele for flower color, then you're going to, and they're codominant, you will end up with red and you will end up with white in your flowers. And how that's actually going to be displayed, I don't know. Maybe you're red with white stripes or white with red polka dots, but you're gonna have red and you're gonna have white in your phenotype. They're both gonna win. Neither of them's gonna lose. Okay, they're not gonna compromise. So you won't be pink if they're codominant. You would be white and red somehow. And maybe there's another gene that determines how that's going to play out. But so in this example on the slides, I have an example again with flower color with a red allele and a white allele being incompletely dominant. So if you're heterozygous, you're going to end up being pink in this example. And here's an example with chickens. In chickens, the allele for black feathers is codominant with the alleles for white feathers. So black and white, neither is completely dominant. They're actually both dominant. And so if you end up heterozygous for black and white, you have a black allele and a white allele, like this chicken right here, you're going to end up having black feathers and white feathers. You're going to have both. And so you end up, in this case, they call it speckled. So that's some examples of different degrees of dominance. Mendel did not have the opportunity to kind of figure that out or observe that that's true sometimes for certain alleles, that certain alleles, when they're different, not necessarily one's going to be completely dominant over the other. And so in your notes, I have a couple practice crosses for you guys to look at involving incomplete and codominance. I'll do a couple of these examples with you just so you can see it's still the basic thing we're going to do when we set up a Punnett square to figure out the possible offspring. But we just have to keep in mind now that when we look at the phenotypes, we have to be careful if we're looking at incomplete dominance or codominance, because that's going to affect the heterozygous phenotype. So here's an example. I'll pull up this sheet on my camera here. I'll do this first one with you. Just in this example, it says in snapdragons, red flower color is incompletely dominant over white flower color. Determine the expected phenotypic and genotypic ratios from crossing two pink snapdragons. So you have a red allele and a white allele, and we're saying that they're incompletely dominant, which is why there's apparently some pink snapdragons. And so this wants you to cross two pink snapdragons. So if there's pink snapdragons, those must be heterozygous. Okay, that's the only way you're going to be pink, is if you have a red and a white allele. So basically in this question, what you're doing is you're going to take a snapdragon that has a red and a white allele, and you're going to cross it with another snapdragon that has a red and a white allele. And so you would set up a Punnett square like you normally would. In this case, we would use a 2x2 Punnett square to show the different way these alleles could be passed on to the offspring from both parents. So here's a simple Punnett square here. And so for this parent, they have a red allele and white allele. They can pass on one, but not both. So when this person, let's say this is mom, when mom's making eggs, in her eggs, she's either going to package in there a red allele or package a white allele in her eggs, but not both. That's the law of segregation, right? You can only pass on one. Same thing with this parent. Either this parent's going to pass on the red allele or the white allele to their offspring, but not both. And so if this is dad is making two types of sperm. Some of his sperm have the red allele, some of them have the white allele. It's 50-50. And so here are all the ways that egg and sperm can meet, and these gametes could join together and these alleles could combine. So you could have this situation where this egg and that sperm combine to form this offspring, who's going to now get a red allele and a red allele from both parents. Or you could have this one right here, or this one right here, or this kid right here, depending on which alleles it's getting from both parents. And so this question wants you to determine the expected phenotypic and genotypic ratios. Let's start with the genotypic ratios. So for the genotypic ratio, you can see here that there's an outcome that leads to being homozygous for red, and there's an outcome that leads to being homozygous for the white allele, and there's two outcomes that lead to being heterozygous, and it's a 1 to 2 to 1 ratio. So we could write that like this. For every one red, there's two outcomes that should be what? Heterozygous. And there's one outcome that should be homozygous for white. That would be the genotypic ratio, 1 to 2 to 1 in the offspring that are produced. Or 25% should be this, 50% should be that, and 25% should be that. Now for phenotypes, be careful. Because when you look at the phenotypes, you got to remember that red is not dominant and white is not dominant. They're incompletely dominant. So this kid who ended up with two red alleles, that would be a red flowered offspring. They would have red flowers. And this kid who has two white alleles, they're going to have white flowers. But these kids who are heterozygous, have a red and a white allele, they're not going to be red and they're not going to be white. They're actually going to be pink because they're incompletely dominant. So you would end up again with a 1 to 2 to 1 ratio for phenotypes. So for every one kid that has red flowers, you're going to have two kids that have pink flowers and one kid that has white flowers. Just like that. And then there's another example that you guys can try on your own. In this case, they're talking about dark brown fur is incompletely dominant to white fur in hamsters. And here you guys can see up here for the alleles, they wanted us to use a C with a superscript. That's very common, especially when you have incomplete or codominance. But then sometimes they just use an uppercase and lowercase letter. Just be careful. Just don't assume that the uppercase letter means dominant, especially if the prompt tells you that it's incompletely dominant. So just keep track of that. But just use whatever letters they give you. It doesn't really matter what letters you're using to represent the alleles. But let me show you an example with codominance. So number 3, it says, in chicken, black feathers and white feathers are both dominant. So this is codominance now, not incomplete dominance. Determine the expected phenotypic and genotypic ratios from crossing a white chicken and a checkered chicken. Is it possible for those two parents to produce a black chicken? So if we have a white chicken, the white chicken would have to have two white alleles. That's the only way you would have white feathers, all white feathers. And we're going to cross that with a checkered chicken. You might be wondering, what the heck is a checkered chicken? Well, since they told you that these are both dominant, then it kind of makes sense. That's probably a heterozygous chicken. That's a chicken who probably has a black and a white allele, and it has black feathers and white feathers. So that would be a heterozygous chicken. And if we cross those, you would set up a basic Punnett square, just like last time, to show the way that these alleles could be passed on. And if I fill this in super quickly without messing up, you get these outcomes here. Depending on what allele each parent is giving, these are the ways it could be inherited in the offspring. And so in terms of genotypic and phenotypic ratios, let's start for the genotypic ratio. We have some offspring that are heterozygous, and we have some that are homozygous for white. And so it's a 2-to-2 ratio, which reduces to 1-and-1. So that's really a 1-to-1 ratio, or 50-50. So we could say for every one offspring that's heterozygous, there should be, on average, one that's homozygous for white. And then in terms of phenotypes, just be careful again, these heterozygous chickens, they're not black chickens, they're not white chickens. They're, in this case, they say that they're checkered chickens. So there's 2 outcomes that lead to being checkered and 2 outcomes that lead to being white. So again, that's a 1-to-1 ratio. So for every one chicken that's checkered, there should be one chicken that's white, or 50-50. And then the last question here says, is it possible for those 2 parents to produce a black chicken? The answer is no. So not possible to produce a black chicken, at least not from that cross between these 2 chickens. They will never, there's no way that they could ever make a black chicken. If they do end up having a black chicken offspring, well then one of these chickens was not the most faithful. But for these 2 parents, those 2 chickens, then not possible. Anyway, number 4 is another example of codominance. In this case, they want you to use a capital B for dark brown and a capital W for white in a different species of hamsters. They're saying that these are codominant. And this time they use different letters. Sometimes questions do this. It's kind of annoying. Usually we like to use the same letter for a gene. This time they're using 2 different letters, even though they're talking about the same gene, but they want to show that they're both dominant. Just go with it. It doesn't matter. Like, you just use whatever letters they want you to use, or pick your own letters. I don't know. But just keep track that those 2 alleles are codominant. And so that's how you do those. Not too tricky. Still the basics that Mendel said, that you have 2 alleles, you're only going to pass on 1. All that's the same. Now, another situation that happens sometimes that is not something Mendel figured out is that there's some genes that have more than 2 alleles available. And that's what we call multiple alleles. Multiple alleles refers to a gene where there's more than 2 versions of that gene that exist. So for Mendel's pea plants, that never really happened. For his pea plants, the traits that he was looking at, the genes that he was looking at, there was just 2 alleles that existed. Like for flower color, for that gene, well, there was a purple allele or a white allele. There was only 2 options out there. You had 2. You either had 2 purple, 2 white, or one of each, but there's only 2 options. Same thing with seed color. You either had green seeds or wrinkled seeds. There was a green allele and a wrinkled allele. Or for seed shape, they were either round seeds or wrinkled seeds, or green seeds or yellow seeds. And so there was only 2 alleles that existed for each gene. But today we know that that's usually, that happens sometimes. For some genes, there are all just 2 versions that exist and you get 2 alleles. But for many genes, there's more than 2 alleles that exist. More than 2 versions of those genes that exist. You still only get 2. That part is true. You only get 2 alleles for every gene, but sometimes there's more than 2 options. And so blood type is a good example of multiple alleles. You guys have an ABO blood type, which is whether you have type A blood, type B blood, or you could have type AB blood, or you could have type O blood. Those are the 4 different ABO blood types. You have one of these. And then you either have positive or negative for your blood type, but that's a different gene. But right now we're just looking at your letter blood type, if it's A, B, or O. And there's a single gene on a chromosome, on a human chromosome, there's a single gene that controls what ABO blood type you have. and you have two alleles, right? You have two alleles for blood type. One your mom gave you, one your dad gave you. That's true. But what's different for blood type is there's three alleles that exist for blood type. There's an allele that codes for having type A blood. There's an allele that codes for having type B blood. And there's an allele that codes for having type O blood. And it has to do with what carbohydrate is produced on your red blood cells. There's different versions of these carbohydrates or these antigens produced on your red blood cells, and it depends on this gene, what type you're making. Now, the interesting thing is, well, first of all, that there's three different alleles that exist. You only get two. Maybe you have two of these, or two of those, or one of each of those, or one of those and one of those. You only get two, but there's three. There's more than two options available. That's what multiple alleles is referring to. Now, the A allele and the B allele are actually co-dominant. So here's an example of co-dominance. A and B are co-dominant. So if you end up with an A allele and a B allele, which is possible, you actually would, that's how you end up with type AB blood. You're going to make the A antigen and the B antigen on your red blood cells. So you're going to have type AB blood. They both win. So that leads to a different phenotype. But the O allele is recessive to both. So if you have the O allele, that's actually going to be recessive to both the B allele and the O allele. So to have type A blood, for example, let's say you have type A blood. If you have type A blood, that means your genotype, you have one of two things. You either have two A alleles, two alleles for A, or you could be heterozygous. You could have an A allele and an O allele because O is completely recessive, so you would still have type A blood. Same thing with B. You could have two B alleles, or you could be heterozygous and have a B allele and an O allele, but B is dominant, so you would have type B blood. To have AB blood, you would have to have the A and the B allele, which are both co-dominant with each other. And to have type O blood, you would have to have two O alleles. If you have two O alleles, that's what leads to being type O, which is recessive. Anyway, you could do some crosses that involve multiple alleles sometimes. Just remember that you only have two alleles, but there's more than two that you might find in people's genes, okay, that exist in the human population. There's more than two options out there, but you only get two. And so when you do crosses, you can get some interesting, some different outcomes now, because now there's more variety in terms of what alleles you have and what alleles you're passing on. So here's an example. I'll pull it up on my paper. I'll do one of these with you. So if you look at number 5, it says, what blood types could the children have if the parents were type A and type B? So one parent is type A, and they specifically tell you that parent's heterozygous. They have an A allele and an O allele. A is dominant, so they have type A blood. And type B, this person's also heterozygous, a B allele and an O allele, but they have type B blood. So you know their genotypes. So when you cross these two parents, you would do exactly the same thing that you normally do. You'd set up a Punnett square. You have two alleles, and you can only pass on one each to your offspring. So you're going to need a 2x2 Punnett square here. So one of these parents has an A allele and an O allele, and the other parent has a B allele and an O allele. And you can only pass on one, as always. So we can look at the different combinations that the kids could inherit these alleles from each parent. And you'd get this outcome here. You could end up getting an A allele from this parent and a B allele from that parent, or a B allele from this parent and an O allele from that parent, or these options over here. And so if you look at the blood types that are possible in the kids, this kid who got an A and a B allele, that kid would have type AB blood. So it is possible for them to have a kid with type AB blood. And then this kid, they could get a B allele and an O allele from mom and dad. This kid would have type B blood. So it is possible for them to have a kid with type B blood. And then this kid got an A allele and an O allele from mom and dad. That would be type A blood. So it is possible for their kid to have type A blood. But if you look at this last option, there's a kid who got an O allele and an O allele from each parent. That would be type O. And so what you see in this cross, is a special cross here between these two parents, where actually they could have a kid with any of these 4 blood types. And there's a 25% chance of each of them being happening when they have a kid. And so that's basically what you do. But there is multiple alleles. There's different alleles, more than 2 alleles. Each one has 2. But that creates a little bit more variety now in terms of what the phenotypes and the genotypes of the offspring could be. And then there's a couple more questions on here that you guys can try out to see if you can figure out. I'll do number 6 with you right now, actually. It says a mother has type A blood. Her son has type O blood. A man comes into their life and says that he is the father of the son. So just to add some drama here, it happens sometimes. It says the man has type AB blood. Is he lying or telling the truth? And so let's see if just based off of our simple genetics knowledge that we learned in high school and blood types, if we could figure out if this is even possible without having to pay hundreds of dollars to order a DNA test. And so let's start with the mom. Mom has type A blood. Now, if you have type A blood, there's two genotypes you could have. You could be homozygous for A. You could have two A alleles. Or this mom, if you're type A, you could have an A allele and an O allele. That's possible. And her son, it says her son has type O blood. So I'll go ahead and write that right here. Son. To have type O, you have to be homozygous recessive. You have to have two O alleles. Okay, so we know the son is type O. Now, there's no question that this is the mom of this son. And so actually knowing that the son is type O tells us which genotype mom has. Because if the son is type O, which means he has two O alleles, one of those O alleles has to come from mom and one has to come from dad. That's how this always plays out. You get one allele from mom, one allele from dad. Now, if one of these O alleles came from mom, that means mom has to be heterozygous. So she must be this, not this. So we now know, just by looking at the mom's son, what her genotype is. So she has type A blood, and more specifically, we know that she's heterozygous because she's going to have a kid with type O blood, and one of those O alleles has to come from mom. Okay. Now this dad, or we're not going to call him dad, let's just call him man. We don't know yet. He has type AB blood, and so to have type AB blood, we actually know his genotype. He would have to have an A allele and a B allele. Right, that would have to be his genotype. So what we could do is we could set up a Punnett square between mom and this man to see what kind of kids they could have had and if this son was possible. You might already be able to easily figure out that this man is telling the truth or lying, but let's make a Punnett square just to prove to someone if they're confused. And so if I set up a Punnett square for this, the mom, right, is heterozygous for type A blood, and the dad has AB blood. So here's the alleles that mom and the man have. And so if they were to have kids, here's what's possible for their kids in terms of what alleles they pass on. Oops. If you look at this, they could end up passing on alleles so that their kid has type A blood. This outcome also leads to having type A blood. This outcome leads to the kid having type AB blood, and this outcome leads to the kid having type B blood. So really, they could have a kid with type AB blood, type A blood, or type B blood, but it's impossible for this couple to have a kid with type O blood, which means there's no way this can be the father. So we're going to say the man is a liar. Right? We don't even need a DNA test. Like, that's not possible. Anyway, there's some examples of that. Okay, another thing that Mendel was not aware of is that there are some characteristics in organisms that are controlled by polygenetic inheritance. Polygenetic inheritance is when there's a certain phenotype that an organism has that's controlled not just by one gene, but by multiple genes. So this would be a situation where there's different genes that you have inside of your DNA in your chromosomes, and for each of those genes you have two alleles. Like, that's true for like a certain gene. Let's say gene A, you have just two alleles for gene A, and then for gene B you have two alleles for gene B, and then gene C you have two alleles for gene C, and the combined contribution of each of those genes is what's going to ultimately determine your phenotype for a certain characteristic. So your phenotype is controlled not just by a single gene that has two alleles, but by multiple genes in your DNA, in your chromosomes, or on different chromosomes somewhere, that each have their own pair of alleles, and all of that information is used to control that one phenotype. And so this would be the way you would know if a trait or a characteristic is controlled by polygenetic inheritance. A lot of times you can tell just by looking at the possible phenotypes for that trait. So whenever you have a certain trait or characteristic in an organism where there's a whole spectrum of phenotypes that are possible, there's a whole range of phenotypes that fall along a spectrum that are possible for that for that trait, that's usually a dead giveaway that that trait is controlled by not just one gene, but by many genes. Because if a trait is controlled by just one gene, and that one gene has two alleles, well, there's only so many phenotypes you could have. But if there's like many phenotypes you could have for that trait, well, then it's probably, or it's definitely going to be because it's not just one gene controlling that phenotype, it's a combination of different genes, each with their own pairs of chromosomes controlling that trait. So a good example is like skin tone. Humans, for example, we have a whole range and spectrum of skin tones that are possible for your guys' phenotypes, from very, very dark to very, very pale, and every skin tone in between. And that's because skin tone is controlled by polygenetics. There's many genes controlling your skin tone. So your skin tone, there's not a single gene that you have in your DNA. There's not just one gene that has a pair of alleles telling you what genotype do you have. Instead, there's many genes, a gene on this chromosome and a gene on this other chromosome and two genes on that chromosome, each with a pair of alleles, a pair of alleles, a pair of alleles, a pair of alleles, and all that information together is what determines your skin tone, which creates a lot of possible outcomes when there's multiple genes involved, each having their own alleles. And so skin tone would be a great example of polygenetic inheritance, along with any other type of trait that has a whole range of phenotypes possible, like height, for example, what height you are. In humans, there's a whole spectrum and range of heights that are possible, from people who are below 4 feet to people who are over 7 feet, and every inch and millimeter in between. And that's because height is controlled by multiple genes, many genes. It's not just one gene inside of you telling you to be tall or short. There's many genes, each containing their own two alleles. Maybe you're homozygous for this gene and heterozygous for this other gene, and homozygous dominant for this other gene and heterozygous for this gene. And you have those two alleles and those two alleles and those two alleles and those two alleles. And altogether, that information is what's going to contribute to determining your height. It says polygenetic inheritance. Mendel did not observe that with his pea plants, but that happens a lot for certain traits. Another thing is the environment can oftentimes impact the phenotype of an organism. So there's lots of traits that can be where your phenotype is affected by what's going on in the environment and not just what's going on in your DNA. So for your phenotype, a lot of times, so far, we've just been looking at, okay, what two alleles do you have? That determines your phenotype. But there's lots of situations where it's not just what two alleles you have or what information you have for a gene in your DNA, but it's also your phenotype is also going to be affected by things going on in your environment. Skin tone, if I just go back to skin tone, that's a great example of a trait that's also impacted by the environment. So in your guys' DNA, you guys do have information and alleles that contribute to what skin tone you're supposed to have. But obviously, as you have experienced so far on this planet, the amount of UV exposure that you have, the amount that you're exposed, the amount of sunlight, for example, that you're exposed to can also affect your guys' skin tone. So there's an environmental component to determining your phenotype. And that's true for lots of different traits. Here's an example with a hydrangea flower. These flowers will change color depending on the soil acidity. So if the soil is more or less acidic, it actually affects the color of these flowers. So these flowers could be genetically identical. These could be exact copies of each other genetically. They could have the exact same DNA and the exact same DNA, which means they have the exact same alleles for flower color as this plant has the same alleles for flower color. It's like the same. But they could end up different colors because of the environment impacting their phenotype and influencing their phenotype. And so the way you know if the environment is impacting a phenotype is, well, one evidence for the environment affecting an organism's phenotype is if you have two individuals who have the same genotype, so they have the same two alleles, this person and this person, or this plant and this plant, yet their phenotypes are different. The only way that would be possible for their phenotypes to be different, even though their genotypes are the same, they have the same DNA for that gene, but phenotypically they're a little different, that would have to be because the environment's impacting and affecting that phenotype. So that's something else that is can make things a little complicated sometimes. And then lastly, the last example of non-Mendelian inheritance on here is something called non-nuclear inheritance. And this is when there's DNA that you're going to inherit that did not come from the chromosomes that are located in the nucleus. And so up to this point in this class, we've been assuming that all of your DNA is inside of the nucleus and the chromosomes that you have in the nucleus. And for humans, you have 46 chromosomes, 23 from mom, 23 from dad, each containing, or altogether containing thousands of genes to produce you, a human being, with all your traits and characteristics and phenotypes. But there's actually some genes that you have, there is DNA that you have that is not part of those 46 chromosomes. If you guys remember from last semester, we talked about how the chloroplast and the mitochondria are two organelles who have their own set of DNA. There's DNA inside the mitochondria, and there's DNA inside the chloroplast, which plants have chloroplast and mitochondria. We only have mitochondria, but there's DNA in there. And it's circular DNA that actually has genes on it. There's instructions to code for certain proteins that are actually used in your mitochondria. Like there's important genes in your mitochondrial DNA. Same thing with chloroplast DNA. There's important genes in there that make important proteins for the functioning of the mitochondria and the chloroplast. Now, that DNA in the mitochondria, at least, is inherited solely from your mom. The egg cell that was used to make you. When your mom was making egg cells, the egg cells that the mom is making through meiosis is packaging a combination of chromosomes in there, but it's also producing gamete, sex cells that have mitochondria. And those mitochondria are going to come from the mitochondria that's in your mom's cells. And what happens is during meiosis or mitosis, when the cell's dividing, it copies its organelles, including the mitochondria, and it will copy the mitochondria and disperse those mitochondria to the daughter cells. And so whatever DNA is in your mom's mitochondria is going to end up being the DNA that's in the egg cells that your mom's producing in the mitochondria that's inside the egg cells. And so here's a picture kind of showing you what I mean by this. So egg cells have mitochondria in it. This mitochondria has DNA, and these mitochondria were copies of the mitochondria that your mom has in her cells that were used to make this egg cell. The sperm actually also has mitochondria in it from your dad. This is mitochondria that your dad has, and that's in the sperm cell. But the thing is, when the sperm cell fertilizes the egg cell, the mitochondria from the sperm cell doesn't end up inside the egg cell. So once this egg is fertilized and the DNA, the chromosomes from your mom's egg and your dad's sperm join together and make a diploid cell that now has two sets of chromosomes from your mom's egg and chromosomes from your dad's sperm. It's also going to contain the mitochondria that was already here in your mom's egg cell when she made the egg cell. It doesn't contain any of the dad's mitochondria. And this cell right here will then start dividing and developing into an embryo and into an offspring that's produced. And all of the future cells that come from this fertilized egg will be using and replicating these mitochondria that came from your mom. And so that means that any gene related to your mitochondria DNA, any genes that are located on your mitochondrial DNA, which there's a handful of genes on there, those genes are going to be the genes that you get for those. I mean, the DNA that you get for those genes is going to solely come from your mom. So you will only get your mom's mitochondrial DNA. Your dad's mitochondrial DNA will never be passed on to his offspring. And so for that reason, sometimes there's like certain disorders that are connected to the mitochondria DNA. There's mitochondrial diseases where the mitochondria aren't working very well. And sometimes that's because of the DNA inside the mitochondria. It's bad DNA. And that would have had to come from the mom, that person's mom. Like that's where they got that mitochondrial DNA from. And so in this picture here, it kind of shows you like there's lots, cells can have many mitochondria, and some people might have a small number of bad mitochondrial DNA. And so these red ones represent like some mutated mitochondrial DNA. These mitochondria might not work very well. And when the mom is making egg cells, it's going to randomly separate those mitochondria into egg cells. And sometimes you might end up with combinations of mitochondria, a bad collection of mitochondria that came from your mom's cell when it was producing egg cells. And if that egg cell is used, which has a high amount of bad mitochondria, you're going to end up with a fertilized egg that is going to develop into an offspring that might have a lot of problems because of the DNA they inherited in their mitochondria that came from their mom. And so the more bad the mom's mitochondria is, the more likely it is that she has a mitochondrial disease, and the more likely that she's going to pass on her bad mitochondria to you, and that her offspring are going to have a mitochondrial disease. And so we'll see, like for these mitochondrial diseases that I'm talking about that have to do with the DNA and the mitochondria, we'll see that it's passed on through the mom. And so if you have a mitochondrial disease, it's usually because your mom had a mitochondrial disease, or sometimes a maybe not so severe version of that disease, depending on how many bad mitochondria she packaged into your egg cell. But yeah, so that would be inherited solely from the mom. So if your dad, for example, if I was born with a mitochondrial disease, let's say, and I have some bad mitochondria, which I would have gotten from my mom, okay, through the egg cell. Well, when I have kids, my kids will be unaffected by that. My kids, just because I have bad mitochondria, I will never pass that on. I'm not able to pass that on to my offspring. It would have to be only the mom who can pass on the mitochondrial DNA. So that's what's going on here. So there are some genes in plants and animals that are located inside the mitochondria and chloroplast, and that's going to be inherited a lot differently than the rest of your genes are, which are located on chromosomes, and you get a set of chromosomes from mom and a set of chromosomes from dad, and you have 2 alleles for it. This is going to play out a lot more differently, mainly that this is going to be solely inherited from your mom and have nothing to do with your dad. So that's it for this topic. Hopefully most of that makes sense. There's questions for you guys to go through and practice. I'll see you in class.